My name is Ekaterina Perishvili. I am a scientist from University of Geneva. I am working in the field of regenerative medicine. Specifically, I am trying to develop a bioartificial pancreas utilizing tools of tissue engineering on organ regeneration. Type 1 diabetes is autoimmune chronic disease which affects mainly children, and current management of type 1 diabetes involves daily injections of insulin that cannot deliver the cure to the patient and is only supportive therapy. Lost function of islets can be replaced either by whole pancreas transplantation or pancreatic islet transplantation, but it cannot be offered to all patients that are suffering from type 1 diabetes, since these procedures are requiring lifelong immune suppression. In VANGUARD Consortium, we are consolidating our forces to develop bioartificial pancreas that can be offered to all type 1 diabetics without need of immune suppression.
It's very exciting for me to work together in the VANGUARD Consortium with different disciplines, including diabetologists, immunologists, specialists in ethics and surgeon, and our aim is together to develop a new treatment for type 1 diabetes. We put together several cells, including insulin-producing beta cells and cells reducing the immune response, and the third cell are cells which are involved in the formation of vessels. We will develop these mini organs which will be transplanted to the patients to cure type 1 diabetes. And according to our preliminary data, we have great hope that we are on the right way to do this, to achieve this aim, and this will probably change the lives of many people having diabetes, when they have no longer the problem to inject insulin on a regular basis, to monitor the blood glucose. And this is really very promising to reach a novel technology for cell-based therapies. So in our group we are working on the development of the bioartificial pancreas by using insulin-producing cells derived from pig pancreas, which of course must be combined with other cells to reduce the rejection after transplantation. Then the next step would be to really demonstrate that after transplantation it has the same effect, and then we can go on to the first clinical trials, pilot trials, whether we can also see this in patients having type 1 diabetes.
Currently, more than 40 million people worldwide suffer from type 1 diabetes. The gold standard for the treatment of diabetes is the injection of insulin. However, a lot of the patients, they need multiple injections per day. The alternatives to insulin injections is the transplantation of either pancreatic islets or the pancreas as a whole organ. And both of these approaches actually help to keep the blood glucose levels in a healthy range. However, the limitations of these approaches are the shortage of the organs, and that you need lifelong medication to cope with the rejection of the organs. The role of Kugelmeyers in the VANGUARD Consortium is providing the SP5T platform technology. This technology allows the generation of size-controlled, uniform and viable spheroids in a large scale. This large number of spheroids is also needed for later translate these ideas towards clinical applications.
VANGUARD is a quite ambitious project in which we want to have a cell product that is able to cure diabetes without immune suppression. To do this, we need to put together many different things, mainly 3 different cell types. One is cells that produce insulin, the other one are cells that make this pseudo-organ, something that is that engraft in our body, and some cells that can induce tolerance, so can avoid immune reaction. This is the great difficult part of the story, because we have to put together different parts that they can work, but they have to be combined in the right way, in the right time, and in the right microenvironment. Our group is mainly dedicated to test this type of bioartificial organs in preclinical model, and to look if it is really able to cure diabetes, at least before using in humans.
Diabetes is due to the high level of glucose in the blood. There are two types of diabetes. Type 2 diabetes, that is due to uncontrolled level of glucose in the blood, but are due to the fact that the patients are not able to use insulin properly. Instead, for type 1 diabetes, there is a problem that because pancreas doesn't make insulin at all. And so for this reason, the possibility to improve the therapies of this disease is very important, because this is a social disease, and the possibility to reduce the cost of this medication will be very important. My group, that is at the University of Piemonte Orientale, is gonna produce two types of cells. One are the BOEC, blood outgrowth endothelial cells, and the second is to engineer with some viral vector and gene editing techniques another type of cells that are called human amniotic epithelial cells, and hopefully we will not need any immunosuppression. This is what we hope to build in the future years.
Vanguard is an EU project funded under Horizon 2020. It brings together 9 partners from all over Europe, research institutions, SMEs and NGOs, who will be working together for 5 years to find the cure for type 1 diabetes.
I'm a transplant surgeon, and as a transplant surgeon I am faced with patients with type 1 diabetes who are waiting either for a pancreas transplant or sometimes for an islet cell transplant. And what I see with these patients is that they experience the frustration of having to wait for a donor to be available, with sometimes very long waiting times, uncertainties about how long they will have to wait. And the ultimate goal of the Vanguard project is precisely to abolish that, to be able to deliver to the patients an off-the-shelf product that would be available whenever the patient needs it, without the waiting time, without the uncertainties related to organ donors. So that would be, of course, a great progress for these patients. It would also open the possibility of having unlimited supplies, if you wish, of a cell therapy product for patients with type 1 diabetes, and open this possibility to more patients than are currently potential candidates for what we call beta cell replacements, which is currently pancreas transplant or islet cell transplant.